Hello, everyone, and welcome to the latest installment of the CDX Engage webinars hosted by Applicati. This webinar is entitled Unlocking the True Value of Multiplexing IHC from Clinical Trial Programs through to IVD Applications. This is an online event designed to bring you with the opportunity to explore the potential and the long-term aspirations of mIHC for clinical trials and IVD applications. To briefly introduce myself, I am Noemi, a member of the CDX Engage team here at Hanson Wade, and I will be chairing today's session where we will be joined by 4 expert speakers from Applicati, Ultiview, and Targos. The webinar today will consist of 3 presentations, followed by a panel discussion and Q&A, where you will be able to ask your pressing questions to the speakers, together with some quick interactive poll questions to let you provide your feedback. Today, we will hear from Will Howat, Senior Director of Antibody Validation and Characterization at Applicati, Angela Vasaturo, Associate Director of Biomarker Strategy and Applications at Ultiview, Oliver Stoss, Chief Executive Officer at Targos, and Keith Wharton, VP Medical Director at Ultiview. I'm sure you will all agree that our speakers today have wealth of knowledge in the field and are well and truly placed to discuss the potential for using mIHC for clinical trials and how to overcome the challenges of getting it ready for IVD use. Please submit all questions via the Questions tab at the bottom of your webinar dashboard. Remember, you can submit this at any time throughout the webinar. We will try to answer all questions in the allotted time, but all unaddressed questions will be passed along to Applicati so they can follow up with them directly. Before handing over to Will to begin his presentation, I would like to first start with a poll to get everyone started and involved. You will have about 30 seconds to submit your answer, and then I'll show you the results so you can see how your response compares to the rest of the listeners joining us today. I will give you around 10 more seconds to vote on this poll. Okay, so I'm going to share the results with all of you now. As you can see, 46% of you said customization of panels, 56 said data analysis, and 79 said standardization and automation. Okay, moving on to the second poll. I'm launching it now. You will have around 10 more seconds to vote. Okay, as you can see from the results, 40% said you're already implementing mIHC in your clinical trials, and 60% said you are not. I'm sure that during the panel discussion, the speakers will have a lot to say about the results. So now, without further ado, I will hand over to Will Howat, Senior Director of Antibody Validation and Characterization at Applicati, to begin with the first presentation. Will, I will bring you on screen now, and over to you.
Thank you, Noemi. I hope you can see.
Thank you, Will. I can see your slides. I'll leave the stage now to you.
Thank you very much, and thank you for this opportunity to talk to you. And I'm going to talk to you today around the value of using multiplex IHC for clinical trials through to IVD applications. So to kick off, I'm sorry, let me just go to the correct slide. To kick off, I know this isn't the most scientific way of doing this, but a quick poll of PubMed determines, of course, that
sorry to
multiplex.
jump in for a second. We still cannot see your presentation, so if you can just exit full screen.
Of course.
We can actually, exactly. I will send that to you. 1 second. Okay, yeah, if you can just now share your screen again.
Apologies for the delay. That hasn't come up on my end.
Don't worry, no worries. We can try that again.
Thank you.
Okay, now your slides are up. Yes, we can.
Apologies again. And as I say, the value of doing this is that we feel that I think all of us know the multiplex IHC is growing in usage. As you can see, it's not a great way of dealing with this data, inputting it from PubMed. I'm sure there's an awful lot of data is missing. However, I think the point remains that even though we're at the tail end of March, there are still we've covered more than half of all of the publications that were in 2020. So we know it's growing. But equally, on the right-hand side, you can see that if you type in the same things and say clinical trial into PubMed, it's dwarfed by the amount of things that are clinical trial. And if you combine these together and use multiplex and IHC in clinical trial, you can see that the numbers are much smaller, but it's also, in the same way, it's also growing. And the reason it's growing and the reason is needed is a great example here from Multiview, which is that there is a complexity of the data set that we're looking at now, not just in PD-L1 space. Although this is a good example of PD-L1, but in all the other markers are available in order to pull that together in a multiplex IHC and understand the milieu of the tumor microenvironment and how that can impact on patients, an impact on that patient and a companion diagnostic to help to drive the pharmaceutical industry and drive patient decisions. However, underpinning all of this in multiplex is the single antibody. And so from my perspective, from Abcam, that's what we bring in a lot of that data set. So we provide provide a lot of validation in all of our processes and through the entire development cycle when we're performing IHC. So we will screen early as we can. And when we're screening on a multi-human normal TMA, a tumor TMA, and some mouse rat TMA all the way through the process. This happens to be the final QC process of the TMA slides to give us confidence that we've got an antibody that truly binds an IHC. But then we've got that extension, and that extension involves using cell lines. So this is a particular example of CD73, where we're using cell lines, and on the left-hand side you can see positive cell lines, so UE7mg as a positive cell line for this marker, and some negative cell lines such as DAUD. But by adding in knockout and adding in those knockout validated clones that we have available to us, in this case you can see in the bottom panel here, the wild type of CD73 and the aligned knockout, again gone through an FFPE and following fixed paraffin embedded mode for that cell line, you can again be happy that you've got specificity of that clone. And involving that gives you the added confidence as you're demonstrating your antibody works. But then you can extend that into tissue microarrays as we do. So, taking 4 different tissue microarrays that are relevant to CD73, staining those up, and demonstrating the quality of the staining that you're getting in and positivity in various TMA cores. But CD73 is not a particularly clinically relevant marker, and we've got that for other relevant markers which are clinically relevant, such as PD-L1. So you can see here 4 different PD-L1 clones, many of which I'm sure as an audience you'll recognize the names of, as they are either in CDX or are aligned with diagnostics. So, again, those clones are being used in a singleplex fashion within clinical diagnostics, but we've extended those as well, and so we can run those in a tissue microarray, in this case a non-small cell lung cancer TMA, to make sure that we've got the relative sensitivity for those clones in an AppCam setting. And then we can also use cell pellets, again in this case the cell pellets from histocyte, to do 3+2+1+ a negative special, and again bring that confidence to our data set to allow us to know that the antibody is binding to the target that we intend it to bind to through the development process and subsequently. So that gives us a lot of data that we can then analyze, and we can add in all that analysis. And here you see two plots, both image analyzed, one on the left, which is PD-L1 and the small cell lung cancer that we showed earlier. The one on the right is a mixed population of those 4 TMAs, whether breast, liver, or others. And you can see and use this to determine the sensitivity of the assay, determine how the population is structuring. And again, it's all part of that confidence level of knowing the target, the antibody is binding to the correct target. And all of these things are needed. And they're needed because when we then bring that in, we need to bring those individual antibodies as a panel into that multiplex environment. It allows us to know that we can do that in a good way to give us confidence that we need for the spatial decryption of the tumor microenvironment. And we need that because we do know that the future of prognostic predictive biomarkers is in that direction. So having those individual markers pulled together with Ultiview to determine that they fit together as a panel gives that solid underpinning that we need to allow that to go through from there and into the likes of Targos when it becomes into our clinical validation set. And that's the combination that we're trying to combine to put together here in this webinar, as well as in this particular talk. And with that, I will end in this kind of short oversight and end my particular part of that and pass on.
Thank you, Will. Thank you for this. I will now ask you to leave the stage. Our next speaker is Angela Vasaturo, Associate Director of Biomarker Strategy and Applications at Ultiview. Angela, please join me on screen now. Thank you, Angela. Thank you, Angela. I can see your slides are up now. I will leave now the stage and all yours.
Thank you, Noemi, and good afternoon, everyone, or good morning. Depends on where you are. Before, sorry, it's on the other way around. I don't know what happened. Technical issues for everyone today. Can you see my screen fine?
Yes, we can, Angela.
Okay, so before moving really deep, talking about multiplex IHC fluorescence, I would like to give a very quick overview of the evolution of the histopathology and the different types of staining that are used to analyze today morphological features, cellular structures, and different cell types. So on the left side, you see the hematoxylin and deusin, which is the most popular use also for routine diagnosis, and really allows pathologists to identify the morphological changes and then determine diagnosis. We talk of cancer, of course, in this case. Then next to it, there is, we have heard a lot about the classical IHC, mainly DAB staining. And this is basically adding in one single protein detection. And it's currently used, for example, for predictive biomarkers like PD-L1 in the clinic. And it also helps identify a single cell population. Like if you use cytokeratin, you'll be able to look at epithelial cells. If you look at CD3, you'll be able to look at T cells, for example. Now, what is really important is that we have heard from Will as well that the tumor microenvironment is really complex. It's not just made of tumor cells or just T cells. It's made of a complex architecture of all those different cell types. And this is where multiplex immunofluorescence is really helping. So you can then identify, detect multiple proteins on the same tissue, and then also have a deeper cell phenotyping, because many times you need multiple markers really to identify a specific cell with a specific function. Then also being fluorescence, you also can detect a higher expression range, and having multiple cell types identified at the same time, you can look at their interactions and also at their spatial distribution. And this is where basically we have developed the technology that we call in-situplex, which really tries to meet all the needs of tissue multiplexing, which I think I tried to summarize in this slide. So one is the high level of multiplexing for the reasons that we have that I have just described, that also go very much together with the identification of complex phenotypes, because depending on the type of markers you choose and the combination of markers, you can identify complex phenotypes and also rare phenotypes based on the co-expression, co-localization of different markers, but also on the differential level of expression of markers like the functional ones. We also try to build something that would be easily compatible with workflows that are already established in the pathology labs. So the assay is carried out in a short time, high throughput. Is just 5.5 hours to perform the staining. So basically you can go from your slide to data in one single day. And what is I left at the last, but it's really critical, is the capability of doing whole slide imaging. Especially when we work with the tumor samples, we know that there is already an intrinsic very high heterogeneity in those tumors. So we really want to avoid the selection of region of interest that would bias the results. So we really like to look at the entire piece of tissue on a slide that would also mimic what pathologists do usually in the clinic. So they look at the entire piece of tissue, right? So we have built initially multiple kits that we call Ultimapper kits that try to answer the basic questions, the basic biological questions in the immuno-oncology space, like are the T cells in the microenvironment activated? Are there any immune suppressive phenotypes like Tregs or myeloid-derived suppressor cells? What about the immune evasion with the PD-L1 expression, or the T cell exhaustion with the PD-1? So I'm going to show you a couple of examples of images and the type of data that you can analyze and the type of answers you can get from using multiplex on a tissue slide. This is a melanoma sample that has been stained with the kit that we call a T cell activation panel, which includes CD3 that you see here in red, granzyme B in green, and being melanoma, we are showing here the SOX10, which is a specific marker that stains the nuclei of melanoma cells. Now, if you zoom on this area in the middle here, you can much better see the presence and the density of the T cells that are expressing CD3 in red. But now you can also start seeing the expression of granzyme B in the cytoplasm of those cells. And if you focus on the middle one, here you see that you have this T cell that expresses high level of granzyme B and is also in contact with the melanoma cells. And the granzyme B is all polarized towards the immunological synapse, so the contact of the two membranes. This is basically allowing us to visualize in a way the activity, the cytotoxic activity of this T cell that most probably is going to release the granzyme B and induce cell death in this melanoma cell. We have another example of staining. In this case is non-small cell lung cancer that has been stained with the PD-L1 kit, which includes CD8, CD68, PD-L1, and cytokeratin. Then you can see here in different colors on the left side. And if you look at higher magnification, here you have CD8 in green that helps identify the T cells. And then here you go to the PD-L1, which is in red. And at this point, you can really look at the compartment where the PD-L1 is expressed. So you can have it expressed on tumor cells that are cytokeratin positive, and you see this pinkish type of staining. But you can also have look at the expression of the same marker now on the immune compartment, which is identified by the CD68 positive cells in yellow that are the macrophages. So again, combining those different markers, you can look at the localization of the cytotoxic T cells, but you can also identify the different compartments that express the same molecule, which in this case is PD-L1. We also recently built another panel, which is a higher plex. We call it IO Immunoid panel, which really covers the key immune phenotypes that you can find in the tumor microenvironment, because it includes the main phenotypic markers for T cells, but also some functional markers that tell you if the T cell is a regulatory T cell or it's an exhausted T cell, together again with the PD-L1 expression on the two different compartments. And what I want to highlight here is that when we work with clinical trials on solid tumors, we very often have, of course, multiple indications. And then we can use those kits really and apply them the same way to all the different type of indications. And here on the right side of the slide, I have this kit applied to different indications in the same run, including lung, breast, and colon cancer. So one last point I wanted to highlight, and I have started this presentation by talking of the importance of the H&E for pathologists. We have the chemistry we use is really very mild and gentle on the tissue that allows us to preserve the tissue architecture and the cellular morphology, which means that we can now perform an H&E staining on the same exact slide where we have performed multiplex IHC. And what this means for pathologists is that when you look at an image like this, if you are a pathologist, for you, it will be very easy to say what is the tumor mass, what it looks like. And if this is a tumor that is infiltrated, most probably you can say there are a lot of T cells in this tumor that you see here. But based on morphology, you cannot really address the function of those cells. You cannot say if they are memory, exhausted, cytotoxic, etc. By adding on top of it and the same exact region, the function of those cells with the profiling with the multiple markers, you now can exactly say which cell is memory, which cell is exhausted, which one is cytotoxic. So having the combination of the two on the same slide, we believe is really highly important and can even better drive decisions in the clinic eventually. And now I just wanted to go briefly touch upon some published case studies. The first one has been published last year by AMFRES, and it really focuses around the accuracy of the PD-L1 test that is used. And they do two very important things in this paper. One is comparing the quantification by the classical scoring that is used by, in the clinic by looking at the classical DAB staining with the digital image analysis. But they also compared this DAB classical staining with multiplex staining. And they did it with two different technologies. One is the in situplex technology of Altivio, and the other one is the Opal TSA. And what they found, what they show is that there is a very high concordance between the manual scoring and the digital analysis. But they also find, and this is very promising, I would say, for the future clinical applications, they found the very high concordance of the ISP technology, so 99% concordance with the classical DAB staining. And when looking at the other technology, they also found a high concordance, which was now 93%. And what is interesting here is that the most discrepant cases were in the range of 0.2% of positive cells, which means that the sensitivity of the assay is really critical when you do this type of studies. And then another paper was also published last year by Katharina Schwarz. This is a phase 1b clinical trial on multiple indications, advanced solid tumors, and it's a small number of patients, but those patients have been treated with autologous myeloid dendritic cells, BDCA1 positive, together with anti-CTLA4 and anti-PD-L1 therapy. And they really found that some patients really showed a partial response and stable disease, and that the promise, let's say, is that this combination could really open up additional paths for those patients that have advanced solid tumors. And also, they applied the classical DAB chromogenic staining for some of the markers like CD8 and PD-L1. And on the serial section, they also performed staining with a multiplex immunofluorescence with the PD-L1 kit, and they were able to show that there was an increased density of CD8 positive T cells and PD-L1 expressing cells post-injection. So this all it's all very promising towards the clinical implementation of multiplex IF. And with this, I'd just like to thank you for your attention.
That was great, Angela. Thank you. I will now ask you to leave the stage. Our last presentation will be given by Oliver Stoss, Chief Executive Officer at Targos. Oliver, please join me on screen now. Welcome, Oliver. I can see your slides are up now. I'll leave the stage all to you.
Thank you, Noemi, and welcome to this third part of the webinar, where I would like to present the CRO's perspective on how to move now the multiplex assays into the clinics. Just one slide, who we are. So Targos is a CRO acting on clinical trials, phase 1 to phase 3 international clinical trials. We are working under full GCP compliance, have been audited by the FDA multiple times, and are, of course, CAP and CLIA accredited. So we have been talking about the use of multiplex in preclinical trials and in research trials, and we are now at the stage where we think that the technology is advanced enough in order to provide it within clinical trials. And the first step of all these three areas that we are now going through is definitely the validation of the assay. So Will has nicely introduced all the strategies of Appcam, focusing especially on specificity and sensitivity, and that's definitely a very, very important step. And Ultiview on their stage are performing verification steps of the complete kit and multiplex kit. However, there is still a gap regarding the clinical use of the assay. It might be a small gap if we are just interested in research use only. It might be a larger gap if we would like to make an LDT according to CAP CLIA requirements, and it might be even a more larger gap if you are going to CE IVD, which all the three items are managed and under the roof of Targos, depending on the intended use of the assay. So the first thing is that we identify the gap, discuss in the project validation plan with the customer what is required, what should be done. And the nice thing at Targos is that the major 4 items that are required in order to set up then a customized assay are under one roof. This is, of course, the laboratory with various instruments. We have the pathology expertise in-house, and we have the development of artificial intelligence algorithms, especially, as I will point out in a few minutes, with the Visiopharm software. And there is a tight ping-pong game, as you can imagine, between these groups until we have a solid, robust assay at the end. And finally, we also have then the design of customized result input interfaces and the presentation of the data that you require at the end under a CRF 21 port 11 compliant LIMS system. If we look to immunofluorescence, the way we are dealing with, and as already Angela has pointed out, we start once the case has been scanned. We start in detection of artifacts, like tissue folds, hools, correction of the tissue detection algorithm. And then we proceed further in detecting the nuclei in a fully automated but customized way, which is then followed by step number 4, where we then detect the signals in the 4 different channels. So 2 are shown here, but as you know, LTView is working with 4 channels, so which are then in association with these nuclei, which allows us then in step 5 to classify the cells as such, to define where a marker sits, and this is then finally combined with all the markers and all channels, which allows us what we call phenomapping. So we are able at the end to classify the cells based on the combination of markers that has been used in the specific panel. And on top of that, we are now, of course, able to provide bioinformatics in order to make sense out of these data. And at the bottom of this slide, you see some examples what customers currently would like to have provided, and what we are going to provide, and what we are providing currently in clinical trials as a readout. And this is the density of course, of a marker in different regions, such as a tumor, the intratumoral stroma, or the peritumoral stroma. We deliver the density of a specific cell type in these different regions. We have been asked to count, of course, the number of cells of a given cell type within a certain distance to another cell type, depending on a cutoff for this distance, which is important in order to make sense out of this spatial relationship that is now able to access within this technology. The classification into immune hot, intermediate, and cold stages for immuno-oncology approaches is possible. We have been asked to compare, of course, on-treatment samples with baseline to see a shift, and also the translocation within cellular compartments, for example, the move a certain marker from the nucleus to the membrane or vice versa, is possible. So there are multiple points of readouts, and we need to define them upfront with the customer, in order to customize then the complete assay, the app, and of course the LIMS system in order to provide at the end these data. If we move now to the clinical trial services, one of the most important item is, of course, the start. And we have learned a lot of things in the past 4 years, because everything that disturbs at the end the automated algorithms need to be eliminated upfront as far as possible. And one of these examples are shown in the bottom, where you see a typical biopsy, and due to the fixation, it gets tissue folds as you see. And we have now implemented specific techniques in order to get rid of these folds and of artifacts. We have invested a lot of time in the training of technicians and special microtomes in order to guarantee a very smooth way of cutting in a very precise manner. And these are all the things that are very important afterwards and cannot be completely abolished once things happen at that first stage. So sectioning, sectioning is getting a very important part in comparison to the manual evaluation, where things are not so critical at that stage. If we move then further to the laboratory, as you have heard, the major instrument for the LTView technology is the Leica Bond. We have both the open and the closed system in-house, depending on the intended use of the clinical trial. Then we move to 2 further items that are now coming on top, and of course makes things a bit more complicated than in the past, but we can be, of course, also more exact. The first step is the scanner. We know that it is not easy. You cannot exchange one scanner model by the other. It's definitely immediately coupled to the validation. approach you're looking for. And the second layer is, of course, then the algorithm, the software that is evaluating your case. And if you are going through the path that we would follow using the multiplex immunofluorescence, so these cases are first scanned using the Zeiss Axioscan Z1, and we have exactly the same profiles as Ultiview. We paid a lot of attention and worked intensively together. And thank you again, Angela, for your assistance in the knowledge transfer here, because we are now able to exchange also experience and also round robin trials with Ultiview to make sure that the required level of validation can be approached. Yeah, and then once the slide has been scanned, the images are saved on a CRF 21 port 11 image database, which is connected to our LIMS system. And we have spent quite a lot of effort in the Visioform software interface to our LIMS, because typically the softwares that you get on the market, they are working very nice, but they do not they are not compliant to GCP and not compliant to the requirements of the authorities. They don't have any audit trail. They don't have any control which app has to be used by which pathologist for which case. So everything is guided here in our workflow by our LIMS system, and the LIMS tells the pathologist which app to be used for which clinical trial sample and what has to be done, and everything is under full control according to GCP. Yeah. The LIMS also then controls all the steps that are still existing in a clinical trial world, all the complexity, not only the technique alone, but also the complexity of the different algorithms of a clinical trial, such as country-specific demands, site-specific requests, sample type-specific workflows, result-specific workflows, the informed consent handling. And this is also managed fully electronically at Targos, where we predefine a customized project, as you can see an example on the right. So once the sample is registered, the system knows exactly at what time point from which person certain activity is expected. Yeah. And yeah, this is just to close here. So if you're interested in going a bit deeper into that, we have a nice booklet for the predictive biomarkers in oncology, with a lot of co-authors and the network of Targos. And I'm now happy to take questions on how to move immunofluorescence and the antibodies of Appcam into the clinical trial business. Thank you very much.
Thank you, Oliver, for this. I will now ask you to leave the screen. Thank you, Will, Angela, and Oliver for your presentations. We will now be moving on to the panel discussion and Q&A. So if you would like to submit a question, please do so in the question box. If it's for a specific speaker, please indicate who your question is directed to. For this panel discussion and Q&A, we have the pleasure of having 4 leading experts. Will Howard leads Appcam's global imaging and immunohistochemistry team as Senior Director of Antibody Validation and Characterization. Prior to joining Appcam in 2018, he worked at AstraZeneca as team leader of its molecular pathology group, where his team developed biomarker assays for preclinical, phase 1 and phase 2 clinical trials. Angela Vasaturo is the Associate Director of Biomarker Strategy and Applications at Ultiview. In 2012, Angela was among the first postdocs in Europe to be involved in the development of multiplex IHC, a multispectral imaging and analysis of up to 6 immunofluorescence markers. And today, she is considered a leading European expert in multiplex IHC techniques, multispectral digital pathology, and tissue imaging. Oliver Stoss is a trained biochemist with 20 years of experience in precision medicine. Since 2009, he is leading operations at Targos, which includes the analytical and clinical validation of novel test methods, their application in prospective and retrospective global clinical trials under GCP, and their launch as complementary or combined diagnostics on the market. He is currently the CEO of Targos in South San Francisco. Keith is VP and Medical Director at Ultiview. Leading Ultiview's assay service laboratory, he brings expertise in investigative pathology, multiplex microscopy and imaging, and digital pathology to the customers striving to realize the promise of personalized medicine. Before joining Multiview, he was Senior Medical Director of Leica Biosystems. So after this introduction, I'd like to invite Will, Angela, Oliver, and Keith to the stage so we can begin with the panel discussion. Please all join me now on stage. Welcome, Will. Thank you for joining me for this panel discussion. Before we start, I'd like to remind everyone that they can submit their questions using the questions tab at the bottom of the webinar dashboard. So I would like to start with a question and get the conversation started. This question is not addressed to anyone specifically, so just please feel free to jump in and give your opinions. So based on my understanding, the current probability of success of a drug reaching the market from initial research is really low, around 3%. So based on your knowledge, how do you think biopharma can improve these statistics? Keith, maybe do you want to start?
Sure. Well, as you know, it's a staged process, right? At the beginning, you get license from the health authorities to even try a therapy in man. What you're looking for at the early stage is any kind of signal that can help you understand what type of patients might respond. I think, you know, even the late stage trials with CDX programs, these often fail because the biomarkers, frankly, are too simple. I think what multiplex immunofluorescence and other multiplex technologies are realizing is that what's needed at the next stage to accurately link the patient's lesion, the patient's pathologic lesion, to a therapeutic hypothesis is a multi-analyte biomarker. So I do believe that proper selection of multi-analyte biomarkers are going to help improve this, the success rate dramatically.
Okay, great. Thank you, Keith. Will, do you have anything else to add?
No, I think Keith did a great job of representing that. I think there's a number of stages as we go through that development process, from preclinical all the way through, of identifying the right biomarker. I think that's a difficult piece to do. And it changes, you know, at that preclinical stage, and often the target, the antibody that you choose, may change as you're working your way through those individual stages to finalize that into the clinical arena, and you know, that takes quite a long time. So, you know, identifying where you're going to be right at the earliest opportunity is the best place, and then, as Keith said, you can put in all the other pieces that you need to bring all the multiplex parts together as early as you can within that process, and get as much data as you are able to from the early phase 1 all the way through into the later stages.
Yeah, thank you. So I've seen some questions coming in, so I'd like to kick off with this one. This is addressed specifically to Oliver. So if pre-analytics are critically important for endpoint downstream analysis, how do you see mIHC making it to the common clinic?
Yeah, I mean, that's the bottom of everything. I mean, Will has already shown how many efforts are required and also necessary in order to make sure the first step, which is specificity and sensitivity. And of course, I understand all doubts that we know that even with one antibody, things are very difficult, especially in the manual world, where we have then also the pathologists with their with a certain amount of lack of concordance on that. And if you are now talking not about one antibody, but 4 or even 8 or more of them, things are getting, of course, complicated. But I think that we have gained a lot of knowledge in the past through the singleplexes, through the behavior of the singleplex technologies. And regarding the Ultiview technology, what is the smart thing is that it is a quite rapid possibility to implement a completely new set into the clinics. And at least in phase 1 first, of course, and early phase 2 in order to observe that. But it's definitely something that is easy to scale up, especially also from the validation perspective of the app development that we do not have to forget, and which also takes quite a long time. So currently the app development at Targos takes longer than the development of the UltiView wet lab activity. Yeah. But still, also here, immunofluorescence is very attractive because we are working with definite channels, which is much easier to treat than with bright field. So I think that if there are of course a lot of burdens. Yeah, we have a lot of antibodies that we have to control. We have additional technologies like the scanner and the software that we have to control. Especially the last two parts are new to the community and will make it a bit more challenging in order to move to the clinical trials, to late stage clinical trials at least. But we see that the technology is getting more and more robust. Yeah. And I think that these are challenges that can be solved.
Real quick, a popular mantra in image analysis world, right, is garbage in, garbage out. And I can't emphasize enough the importance of standardizing pre-analytics as a foundation to make technologies like multiplexing applicable across clinical trials and large group. Quick shout out to the efforts of the Anatomic Pathology Patient Interest Association group I was affiliated with prior to joining UltiView, who's strongly advocating for standardization of pre-analytics for all cancer biopsies, in line with the efforts of Carolyn Compton, formerly of the National Cancer Institute, but a strong advocate for standardization of pre-analytics. In the US, it's only breast biopsies that are required to undergo strict fixation times and transport times in line with CAP-CLIA lab accreditation. Other types of tumors are not required to do that, and I think implementing these kinds of strict standards and linking them to lab accreditation is going to be critical to creating materials that are even worthy of subjecting to these really powerful multiplex technologies, whatever they are.
If I could add a very quick question to Keith, is there a role for, as we extend panels and multiplex, a role for a surrogate marker to identify, partly as a structural marker, identify where there's poor fixation present? Is that feasible?
So there's no perfect marker. As you may know, some customers like to do a Ki-67 as a rough estimate of tissue viability. Histology itself, the appearance of the tissue is an adequate measure, but it's a relatively late marker of tissue degradation. There was a large study published a couple of years ago looking at autopsy tissues post at basically the post-mortem interval prior to fixation of tissues, and looked at hundreds of genes and the effect of delayed post-mortem interval on the abundance of those genes as transcripts. So I think the field is a long way from standardizing this, but at least now we have the tools where for specific situations we can do a bit of forensics to understand, for example, what the robustness of the sample quality is, and actually set those standards and insist that every patient have biopsies collected under those standards, and really not waste technology on samples for which we know the results are going to be poor.
But I mean, one thing is true, if we are talking about 4 antibodies in a panel or even 8, and we would like to move to LDT, where Targos is working on with certain customers, we need the effort of testing of the stability of the antigen at the end is of course multiplying. We cannot omit that, especially because its combination is customized, but that's doable. Yeah, I mean it's a matter of planning and the right controls at the end to be employed.
I think 2 features there are important. One is that the system be stable as defined by the use, and that you also have the documentation to support that. And certainly the IVD designation requires you to generate that data, but the predicate to that, or a prior need, is to make a very robust reagent and staining system.
If I may add a little thing, I think it's really critical at this point, the combination, like the combined effort of different companies as well, that can really take care of the different aspects of this process that is a long process to standardization.
Thank you, Angela. So another question we have here is, all biomarkers need to go through clinical trials, and many are single agent biomarkers. How do you select a biomarker combinatorial from multiplex IHC for this purpose? It's not addressed to anyone specifically, so.
Maybe I can comment on that first. I mean, these images that ISP and other multiplex technologies create are beautiful, right? They could be in art museums, but that's not why we generate them, right? We generate them to generate data, and with the combination of markers, as Angela mentioned, we use it to identify existing and newly emerging cell types that are of pathogenic importance, right? In the tumor microenvironment, they're either cells that promote, broadly speaking, they're cells that either promote tumor growth or oppose tumor growth. And the ideal therapy would attack all the cells that are promoting tumor growth and oppose the cells that are opposing tumor growth. Did I say that correctly? I think you know what I'm saying. So I think that all of this data, and I think the standardization of the data and the methods needs to ultimately converge on the cells, the cell types, the agreed upon cell types and marker combinations that represent those cell types, so that we can begin to standardize the readouts of these multiplex methods. I think there's a lot of different antibody sources for each marker. There's different microscope manufacturers for scanners. There's different optics. There's different detection methods. But all of this ultimately has to be linked to some sort of a gold standard. Currently, the gold standard is DAB IHC. It's a good gold standard in some ways, and in some ways it's not. But I think what we have to converge upon is standardization of the recognition of specific cell types and their role in disease. And this relates to ultimately a therapeutic hypothesis held by a pharma company, and that justifies the investment in testing in humans of a specific drug that targets those cell types or some function of those cell types.
Thank you, Keith. So we have another question addressed specifically to Angela. So Angela, the SOX10 uses light blue, quite close to DAPI to my bare eyes. Is the computer powerful enough to differentiate the two wavelengths?
Well, actually, I haven't mentioned the different fluorescent channels that we use, and the colors you want to use to display them are really up to you. Those are pseudo colors. You can choose whatever you want. But in that specific case, the SOX10 was in the size 7 channel, which is quite far from DAPI. And we usually use the classical 4 fluorescent channels that are spectrally distinct, and that with a classical microscope and the classical filters, you can easily separate, and you don't need spectral mixing. So the short answer is yes, although we have made it in light blue, this was not really light blue, and you could have put it in white or yellow or whatever other color you would have chosen.
Thank you, Angela. So then I guess a question I have for all of you is, what level of scientific validity do you believe is needed for a successful clinical trial? Like, how good is the evidence today for the association between the biomarker and the clinical condition?
Just a quick comment on that. I would say most of the evidence generated so far is based on retrospective cohorts. So, large number of patients with X disease were, let's say, selected with PD-L1 IHC or not, and the tissues generated, and there's extra tissue, or there's exploratory objectives of the trial that allow additional investigation. And typically, a typical phase 3 registrational trial, could be thousands of patients. The workflow here is getting faster with multiplex, but it's still relatively slow relative to the standard IHC workup. So I think the data and the confidence to de-risk large programs leading to IVD designation has to come from retrospective studies. It has to come from small-scale pilot studies done either by forward-thinking central labs like Targos, or from academic medical centers, cancer centers, and the like. Cancer is so blazingly complex that we don't know all the ways in which we can begin to cure it or improve outcomes. And so we need to do a lot of experiments with a lot of different methods and a lot of different, frankly, a lot of different ideas. The strength here is in diversity of approaches.
Thank you, Keith. So we're running out of time, so I just want to ask one last question I've seen coming up. So do you see mIHC making it to CE-IVD or acting as a tool to define the few biomarkers to get to CE-IVD? If the former, which indication is the closest to needing this technology?
I absolutely see that it's gonna reach there, and the work that's being done, world of you and leading into targets just helps to pave that way ultimately. As you go through all those testing regimes, as you lock it down, as Oliver showed in his talk, to make it in a compliant way, you can see that's the data set that is required ultimately for then putting out their registration. So yeah, it's gonna be that. It's just a matter of continuing to push those through, from all the way through the entire process, as Angela said, from the original antibody up through AltiView into Targus' hands to lock it out and put it through clinical, large numbers of trials. That's my view.
Thank you, Will. So as I said, we're running out of time, so all unaddressed questions will be passed on to Appcam and they will follow up with you directly. Before I ask you to leave the stage, I'd like the audience to vote into more other polls and maybe get your opinions on the results obtained. So I'm going to share that poll right now. You'll have around 30 seconds to vote. I'll give 10 more seconds. And before sharing the results, I'm going to open the last poll. So there you go. You'll have around 30 seconds more to vote. Okay, thank you. So before the panelists leave our stage, I'm going to share the results, and if any of you want to comment on the results. So I don't know, maybe Will, do you want to start?
I'm just looking at the results. So that's interesting that there's obviously there's a group of people that it's still too early, and I think that's great, and I'm happy to work with you to work that. And what else do you need to work with? Great that there's some IVD use in there, which I think to reach out to the people on this panel and reach out to us to try and help to stimulate those discussions and get you to where you need to be.
Thank you, Will. And I'm going to show the results for the last poll. So for those who answered yes to this last poll, just email us directly at info@hansonwade.com and we will put you in direct contact with Appcam. Great. Well, thank you again for your presentations and for the panel discussion. Please feel free now to leave the stage.
Thank you very much.
Thank you.
Thank you.
Thank you. Pleasure.
Bye.
That is all the time we have, but I hope you enjoyed the session. All unaddressed questions from today will be passed on to Appcam. If you do have any follow-up questions or comments, please email this to myself at info@hansonwade.com and I will get back to you soon. On behalf of Appcam and CDX Engage, thank you all for your time and I hope you enjoy the rest of your day. Hope to see you next time.